So today we're going to talk about introduction and overview into biology for cell biology or biology 160. So biology is the study of life, and the way this textbook likes to present things is through the eyes of evolution, looking at how processes have changed over time on Earth. So when we look at things that are alive, there are some properties that are associated with living things that we want to consider. So living things are always going to have an ordered structure. Things are not random. So when we look at this sunflower here, for example, it's not random where the petals are around the outside. Things are in order and every flower is going to look similar. There's evolutionary adaptation. Things are adapted to the environment that they live in. They're also going to respond to the environment. So it's showing here a Venus flytrap responding to the stimulation of an insect landing in it. If something changes in the environment, a living being is going to react to that. Living organisms are going to reproduce. So we can see here with the giraffe and its infant. There's metabolism. This is how we are going to acquire energy, as well as how energy is processed throughout a body. And then living things are going to grow and develop over time. So when we look at biology, the study extends from all the way down to the microscopic scale with things you can't see to the entire living planet. What we see is there's novel properties that emerge at each level as you go from things that are more simple to more complex. So there are new arrangements, new interactions that are going to make the things as you go up the hierarchy more complex. When studying biology, a lot of times we'll use reductionism. It reduces the complex systems into simpler parts to study it. And while this works to make things easier to study, it does have limitations in there because it can't fully explain the higher levels or the interaction of different parts of the body. For example, with anatomy and physiology, we tend to study things by systems, but those systems definitely interact with each other. And so the sum of the parts is, the whole is greater than what you get from just the sum of the parts in there. And you need to look at overall as well. With Watson and Crick, molecular biology was studied looking at the structure of DNA, but they didn't actually study and explain any interactions. They weren't understood it then. So we have to balance reductionism with a more holistic approach of looking at things. So systems biology just attempts to study these components that function together. So something to keep in mind is organisms are going to interact with their physical environments. So we have cycles that just continue to interact with each other that we see in living things. So water and minerals for the soil are important for plants. The roots from the plants will actually help to break up the rocks, which are going to release more minerals into the soil. So it's a continuous cycle. Carbon dioxide gets taken into plants and they're going to release oxygen. The animals are going to take in the oxygen and then release carbon dioxide. So while we tend to hear carbon dioxide is a bad thing, it's not actually. It's an important part of the cycle. Without carbon dioxide, you would not have plants. They would have nothing to eat. Insects and animals are going to eat plants, and then when they decompose, they're going to return those minerals and nutrients back into the soil that the plants will then consume. So the ecosystem really has multiple cycles that are just continually repeating themselves. So when we look at the transfer and transformation, the producers are going to be the organisms that are going to absorb light energy and transform it into chemical energy. These are going to be the plants. So they are essential for everything else because the consumers are all going to feed on those producers. They're going to make the organic products that the other consumers need. So it involves a transfer of chemical energy from the food of the producers to the consumers. What the consumers use is going to end up being used for doing work and producing heat. So when we look at structure and function, they say form fits function. the anatomy of life fits its function at all structural levels. So all anatomy is going to have a function. There isn't parts that are out there for no reason. We may not understand the reason, we may not understand it well, but so far everything that we have does have a reason for that part being there. So when we look at the basic unit of structure and function, it is the cell. There are some characteristics that all cells share. They are enclosed by a membrane. That membrane is going to regulate what goes in and out of the cell, and they're going to have DNA as their genetic information. Cells are going to come in two types, eukaryotic cells and prokaryotic cells. The eukaryotic cells are going to have internal membranes that will separate the nucleus and the organelles. With prokaryotic cells, the DNA is not separated by a membrane. They also do not have any membrane-bound organelles. We see continuity in heritable information. So things are going to be continually passed down from one generation to the next. The DNA, or deoxyribonucleic acid, is that genetic information that's going to pass down from one generation to the next. So your genes are made up of DNA. They are the unit of inheritance that will code for a protein. So looking at DNA's structure and function, what we typically picture is a chromosome, which is a long strand of DNA that's going to have hundreds of genes on it. Usually we're only going to see the chromosomes be present during cell division. The rest of the time, the genetic material is going to be chromatin, where it will be more loosely coiled and enzymes can get in and access it. The genes are going to provide instructions to build the proteins. The genes are on the chromosomes, on the DNA. Proteins are going to be what creates structures. They're going to carry out the cellular work and the cell's identity. So the function of the chromosome is to replicate when the cell is getting ready to divide. Then each of the offspring will get a complete set of the genes when it inherits those chromosomes. So that DNA is the central database, or the central set of instructions for everything that organism does. When we look at the molecular structure of DNA, it's a double-stranded helix. So it's like a ladder that's been twisted. There's 4 nucleotides, A for adenine, T for thymine, C for cytosine, G for guanine, that are going to make up the rungs of the ladder, and then it's sugar and phosphate that are going to make up the sides. Their specific arrangement is going to code the information in the genes. So where we hear about computer codes, it being a series of zeros and ones, this is just going to be a code that involves A, T, G, and C. So gene expression is when the DNA of these genes is going to control protein production. So indirectly, it's going to use an RNA intermediary. The DNA is going to be your long-term molecule that's protected, but the RNA will provide a working copy that will go out into the cell, and that will direct the production of cellular products. So the genome is the entire library of genetic instructions that an organism inherits. Genomics just studies the sets of genes and compares the genomes of the species rather than a single gene like genetics does. So with genetics, we've rapidly acquired vast amounts of information here that are tedious to process. And so with high throughput technology, we're able to use tools to analyze biological materials. So a lot of this is computerized. Bioinformatics just uses computers to store, organize, and analyze the data. So if you were to think about it, if you were to be given a 1,000-page document and you needed to go in and highlight the word "the" in there manually with a highlighter on a piece of paper, it would take you a long period of time. Where on your computer, you can just simply search for that in a PDF file, and with a matter of a couple seconds, it's going to come up with a document that has that word highlighted in it every place for you. So computers have made it possible to really rapidly analyze vast amounts of information. A lot of times research teams are interdisciplinary. We have diverse groups of specialists working together. You can't be an expert in everything. And so having these interdisciplinary teams allows people to be experts in their field and to be able to have the benefits of working with other experts. So one of the common things we see is regulation through feedback mechanisms. In a living system, we refer to this as homeostasis. When you attempt to keep a constant or stable environment, you can think of it being somewhat like a thermostat in a house, where if it gets too warm, it's going to turn on air conditioning. If it gets too cold, it's going to turn on the heat. So if you're in an old house and you don't have air conditioning, that's what newer houses have, that will allow a constant environment in there. So with negative feedback, this is what you see most commonly in living systems. They detect a change and they attempt to reverse it. They say you're getting too far away from this middle of the line here. One of the ways they can do this is you can have the accumulation of end products. So we can see that here where we go through and with a series of enzymes we make product A into B, B into C, C into D. And if D starts to build up in there, it's going to shut down the enzyme between A and B so that the process does not continue on. It will shut it down because it doesn't need more. Positive feedback is less common, but with that, it will detect a change and it's going to reinforce it. So those end products will speed things up. So here, where you're going from W to X, X to Y, Y to Z, as Z starts to build up, it's going to stimulate even more production of Z in that process. So evolution attempts to explain the unity and diversity we see in organisms. So the organisms today are modified descendants of common ancestors. So far, we have identified about 1.8 million species. There's thousands identified each year. We don't really know how many there are. We estimate between 10 and 100 million. Taxonomy is how we name and classify these species, and it's based on shared characteristics. So going from the most specific to more general, you've got the species, genus, family, order, class, phylum, kingdom, and domain. So something to keep in mind later on when you're looking at things, this taxonomic system was developed before we even knew viruses existed, so they don't really fit into this, but viruses are not generally considered to be alive either. So we have 3 domains of life. They're based on DNA sequences. You've got the bacteria, the archaea, and the eukarya. So the bacteria and the archaea are both prokaryotic cells. The eukarya are going to be eukaryotic cells. So the archaea, they have nearly as many similarities to the eukarya as the bacteria. So even though they are a prokaryotic cell, they do have a lot of similarities to the eukaryotes. The eukarya have 3 domains of multicellular organisms, and then some that are single-celled, and they're distinguished by their mode of nutrition. So with animals, their food is going to be acquired by ingestion. Fungi consume dead matter. Plants are going to produce sugars from photosynthesis. And then we have the protists that are single-celled organisms that are very diverse, and they're split into several other groups. The unity we see, some examples would be the genetic code. It's the same used by everybody. There's a few exceptions in there where they will have slight modifications, but otherwise the genetic code is nearly universal. We also see similar skeletons in vertebrates. So if you look at the number of bones in the spine in a vertebrate, it's 24 for everybody. Whether you're a giraffe, whether you're a human, whether you are a turkey, 24 of them. So Charles Darwin is the one that is credited with his work on evolution. In 1859, he published On the Origin of Species by Means of Natural Selection. He was not the only one involved with that work. He's just given most of the credit for it. But in his book, he makes 2 main points. The contemporary species arose from the succession of ancestors, so we say descent with modification, and this happened by the mechanism of natural selection. So his key observations were that we have individuals in a population with varieties of traits, and those people or populations can produce far more offspring than what will survive to produce their own offspring. And then those species are adapted to their environment that are more likely to survive, are going to be more likely to produce offspring, therefore passing on those traits. So that idea that populations produce far more offspring than what survive to produce their own, if you're thinking of it in terms of humans, we don't produce a lot of offspring, so we have a much greater survival rate. But if you look at something like insects, a lot of times they will produce hundreds or thousands of offspring, but not many of them make it. So natural selection just looks at the natural environment and selects for propagation of certain traits in the population. What will happen is the cumulative effects could cause the species to give rise to two or more descendant species. So when we look at this picture here, you've got this population of these insects with a variety of inherited traits. Some of them are darker colored, some are lighter colored. They live on a dark background. So the ones that are lighter colored are easier for a bird to eat. So what that means is more of them are going to be eaten and not survive. The ones that do survive are going to be darker colors. So what you're going to see is increased frequency that those traits that enhance survival are going to go on to live and have reproductive success. So when we look at science, there is the scientific method of study and inquiry. Science is just trying to understand the natural world, and inquiry means we're searching for information and explanations. So we do have a process for doing this that's going to involve some pretty standard steps so that we can try and make it more universal. Let's see if I can get this to scroll smoothly here. So in science, we make some observations, then we make a hypothesis, and then we test it. Science is always questioning, so there's been questioning in science from day one. If there wasn't questioning, we would still be looking at the Earth saying it was flat. So we use observations to gather information. So in here it says that we use the senses. Tools are used for this as well. There are some things that our human senses are not going to pick up, such as radiation, that are very real. Then we record the data. The data can be qualitative or quantitative. Qualitative is going to be numerical, or qualitative, is going to be descriptive rather than numerical. Where quantitative is going to use numbers and measures. So quantitative data is generally easier to work with. We can all agree that 2 + 2 = 4. We can count something and agree how many there are. If it's giving a description of a color or color changes in there, those are a lot more subjective. So using reasoning, we have inductive and deductive reasoning. With inductive reasoning, you derive generalizations from a large number of specific observations and then apply those generalizations to specifics. So for one example, I could be looking at a bunch of insects in here, and I look at it and I say, well, you know what? If it flies, it's an insect, because I saw several hundred organisms in there that could fly. That would be a case where inductive reasoning could mislead me, because when I start to look at the bigger picture out there, I'm going to see there's other things out there that can fly, like birds. With deductive reasoning, you're going to try and understand the big things and then explain the little details from that. So during the scientific process, after we've made some observations, we form our hypothesis. So the hypothesis is the tentative answer to a well-framed question. It is not a random guess. It is looking at what's going on in the observations and then using some deductive reasoning and logic to try and explain it. And the hypothesis is then, well, okay, let's go ahead and test this. So in our example here, we've got a person who is observing that their flashlight doesn't work, and she starts to wonder why. So the first hypothesis is that the batteries are dead. So what she predicts is that if she replaces the batteries, it will fix the problem. So she does that and tests it, and you're able to see that the test falsifies the hypothesis that it does not work. So science does allow for modification of things. So when we modify things, it isn't that we just pretend the other didn't happen. It's always documented in there. But when the first hypothesis fails, we're just a step closer. We know that is not the answer. We can try something else. So here's hypothesis number 2, that the bulb is burnt out. So she predicts if she replaces the bulb, it's going to fix the problem. Goes ahead and replaces it, tests it, and it does not falsify the hypothesis. So here it's going to support the hypothesis that it's true. Not everything can be answered by science. So sometimes people have a tendency to want to use science to explain everything, and there are places where it just doesn't. It has to be testable. If you can't test it, then science is not going to be able to explain it. Has to be falsifiable, so we have to have a way of revealing that an idea is not true. There are some things that just inherently we cannot do that with. Things like supernatural, religion, and personal faith are outside the bounds of science. Science does not say they are not true or that they are true. It just says we don't have the tools to explain this or test this right now. So with the scientific method, this is an idealized process of scientific inquiry. It does allow for some flexibility, but by having an idealized process in there, we can have some consistency in how we test for things. So usually when this is done, you're going to create an experimental group and a control group. An experimental group is going to be the one that's going to receive the test or the manipulation, where the control group does not. This is used as a basis for comparison. So when we have these two groups in here, we consider it a controlled experiment. The variables are the factors in the environment, and you want to minimize the difference in the variables as much as possible. So we can have multiple variables, but when there's multiple variables, you're not going to know which one was actually the cause of the change or which one is actually associated with the change. So we have the dependent and independent variables. The dependent variable is going to be what you're looking for to change. The independent variable is going to be what you manipulate. In science, a theory is going to be something that is going to be well tested and supported by a great body of evidence. This is different than everyday language. Everyday language implies a lot of speculation, but in science, a theory does have more evidence behind it. So in science, a lot of scientists work in teams. It's important to be a good communicator in doing that so that you can work cooperatively, not having to try and be an expert in everything yourself. So if someone likes to work with data a lot, they would have a job in science. Somebody who does not like to work with data but likes to be more hands-on conducting experiments, they can actually have a role as well. There are people that are going to be more involved with the computer aspect of things, somebody who writes software. So you don't have to be an expert in all fields. In science, we try to build on the work of others. If something is well established, we will typically look and say, okay, what can we go further on this? Rather than once you have studied it several times, we don't necessarily try to keep restudying it. So for example, we wouldn't continually study the effects of Tylenol on pain. We've studied that for a long time. It's been around for a long time. What we want to do is build on that work and maybe look at other things that influence pain or other effects that Tylenol might have, but not necessarily what we've already done. The advantage to having multiple people working on things is you have other people to ask questions and view things from different angles. So while we might assume that we would catch everything that is off, all you have to do really is take a look at something that you have written and look for typos and misspellings in there. Most of the time, if you're going to go through and you're going to try and find those things in your own work, you're going to miss them. You're going to look at it and you go, okay, I got everything. As soon as you hand it to somebody else, they will find them in there because they're viewing it from a different angle. They don't have already a bias of what they're expecting to read like you do. So having multiple eyes look at things is always a good thing. Science and technology are very integrated. Science is aiming to understand the natural phenomena. Technology is the application of science for a specific purpose.